The Arnhem Land Plateau, which is in the sort of peach color, was generally sort of excluded because it is sandstone country escarpment country, and it generally contains communities and ecosystems that are of different slopes and drainage types. So how do we go about defining a reference ecosystem? Luckily enough, Sarah has already done that for us through the restoration standards. We can draw primarily from principle 1, and even though there's 6 different principles, principle 1 probably best encapsulates what we're trying to do at Ranger as far as defining a reference ecosystem. And that is that ecological restoration practice is based on an appropriate local indigenous reference ecosystem. So if we drill down and sort of underneath that definition, it says that a reference ecosystem is a model that's adopted to identify the particular ecosystem that is the target of the restoration project. This involves describing the specific compositional, structural, and functional ecosystem attributes requiring reinstatement before the desired outcome, i.e. the restored state, can be said to have been achieved. So now I point at that as compositional, structural, and functional words because that's sort of going to be a focus of the talk going forward. So we'll look just a little bit closely at some of those key areas on the Sarah wheel that they've identified for restoration or for effective restoration. And I'll just sort of overlay those with a number of sort of high-level words and phrases that we've used and plan on using in the future and are adopting to as a tool set to go about monitoring and defining this reference ecosystem through time. So for example, for species composition, we're using things like veg surveys, fauna surveys, and drone mapping. Structural diversity includes things like omics, fauna surveys, drone mapping, external exchanges using omics and bird acoustics, et cetera, et cetera. So we're sort of overlaying multiple different tools for each of those. And this is not an exhaustive set. There are other tool sets that we have and that we'd like to use to try to quantify and define the reference ecosystem. And like in all of those tools, we like to put it in the context of a multi-scale approach from genes to landscapes. So that's sort of omics all the way up to drone mapping and beyond. So specific to this talk is our approach to defining a reference ecosystem. This talk doesn't stand alone. I strongly suggest or I hope that you get some other perspectives on how we've gone about defining this reference ecosystem. Peter Erskine's giving a talk this afternoon about fire history and historical perspectives and historical data. Lorna is giving a talk about the process of how we went about selecting sites. That's tomorrow. And Jay is going to talk about sustainability and resilience and how we measure that in these ecosystems. Right here, right now, for me, we're going to talk about veg surveys and drone mapping. So what was our process for doing our veg surveys? We established 1 hectare plots. There's significant amounts of data that's been alluded to. They're mostly historical 20 by 20 meter plots, which have been a great source of information, but they're somewhat inadequate for structural characterization of the ecosystem. If you were in Nick's talk earlier before lunch, they talked about the Ozplots method and how it's been rolled out across the continent. They have something like 800 or 900 plots that are actively being used as reference site and reference ecosystem characterization. We gladly have borrowed their methods. Gives us in those 1 hectare plots, we get a complete overstory census. We get an understory transect survey, as well as soil sampling for biophysical characteristics. As far as our site selection goes, I won't give anything away from Lorna's talk, but we used a randomly stratified survey. It was stratified mostly by fire history. Slopes were only selected sites or regions or areas that have slopes similar to the final landform design. And we have a proximity measure to only include sites that are within 10 Ks of the mine. The sites have to have a lack of contemporary disturbance. I point out the word contemporary because, as has been stated, these ecosystems have been inhabited and used and you know, have evolved in conjunction with 65,000 years of continuous habitation. And then adding on to that, we have a multi-temporal drone data capture scaled out to tens and hundreds of hectares, sort of surrounding each of these reference sites. So here's just a map overview of the mine and disturbed footprint and that surrounding reference ecosystem. It's bisected by riparian areas and creek lines, but in between those areas are general savanna woodlands. Here's just some quick snapshots taken from those sites. That's what savanna woodland looks like in and around Ranger. There's obviously strong seasonal components between the wet and dry, pre and post fire. All of our surveys have been completed at the end of the wet season before any fire or annual fire. I know teens sessions going on next door in arts and restorations, and I know none of us, we can't be there. So this is my attempt at some abstract art and what an Oz plot should look like. So this is the overstory results for each of those plots. So each plot's a 1 hectare plot, 100 meters by 100 meters. We have 27 different genera that's been identified. So for each color, that represents a tree. The size of the point is the size or the DBH of that tree. Really the take-home message is that some plots are very thick in terms of density, some plots are thinner, and that there's just a few main colors that are represented. So though there's 27 different genera, you can see that the turquoises, which are the eucalypts, are dominant in a fair few of the plots, along with some acacias and a few other corymbias, etc. I think, which are in the sort of brownish green. But I think they give you a bit of an idea of sort of the patterns that exist in those plots. Same with the understory. This is just transect data. So there's 10 transects from each of those plots. Sorghum, which is our most prolific annual grass, is in the sort of gray circles. So there's 1,000 points for each plot. The gray circles are typically the most dominant, but there is a rich understory. It's obviously patchy. Some plots look different, but you know, there's still that sort of take-home message of there's some complexity there, but there are also some dominance that potentially could be used in a restoration context. When we take those plots and we sort of look at some of the structural components and plot them out, you can see here we have on the top left, we take understory cover and plot it against overstory cover. And there's a general negative correlation between the two. It's not exceptionally strong, but there's obviously only 10 plots in this case. Overstory diversity versus understory diversity, i.e. richness. It's a bit of a scatter shot, but you can have high diversity in an understory and overstory, or vice versa, or you can have low diversity. It's not as if each plot is exactly similar to the previous plot in terms of its richness. Overstory cover in the bottom left versus basal area. That's not a surprise. More cover, more trees, greater basal area. It's good to see. But alternatively, stems per hectare versus basal area, you can see is there's a bit of a two-way road there. So you can get high basal area by having lots of stems, or you can have a high basal area by having fewer stems and just larger trees, few large trees. So moving on to sort of the drone side of things, I point out this paper. This came out a few years ago by some people in the U.S., James Kellner. It's called the Case for Remote Sensing of Individual Plants. And what this paper does is it sort of takes a look at our ability to use remote sensing tools to measure individual plants. And in the case of restoration, what are we after? We're after things like measuring ecosystem processes, community structure, demographics, and potentially individual plant health through time. So if you look at the different potential tools we have satellites, we have manned aircraft, we have drones, and we have sort of ground-based systems like terrestrial laser scanning. And drones check all the boxes. You can measure individual plants, you can measure demographics, you can measure community structure, and potential ecosystem processes. So what does that look like for us? We fly a drone over our savanna woodland. we have multiple sensors. We have LiDAR sensors. We have multispectral, hyperspectral, and color imagery. From those sensors, we can measure ecosystem processes, things like we can measure respiration and transpiration rates, photosynthetic activity, et cetera, the types of things that people measure that are ecosystem scientists. Additionally, we can measure structural components of that ecosystem. So we can make a canopy height model. We can say, okay, what's the tree height distribution across this ecosystem? We can drill down another layer. We can measure individual tree metrics, track growth and demographics through time. So we can pick out individual trees and we can return to those trees in a remote sensing fashion. We can look at the timing of fruiting and flowering, and we can take the whole system back down sort of to look at things like foliar chemistry. So now we're looking at individual trees, looking at foliar chemistry. So instead of those ecosystem-wide processes, we're sort of we're decomposing it down to individual trees. So what's our current status there? We're scaling up. We're moving from those 1 hectare sites to what we call whole site monitoring. We're coupling ground-based surveys with multi-rotor drones that can fly sort of 1 to 10 hectares effectively. And then we're scaling up with larger drones, which are vertical takeoff and landing drones that can sort of cover 50 to 500 hectares at a time. What does that look like in terms of operationally, in terms of structure? So we can scale up with it. This is our using LiDAR. So this is just an aerial image of one of the plots. The center image is the results from a LiDAR survey. It's color. The redder colors are taller, green is lower, blue is the ground. From that raw data, we can segment out individual trees. So we can say, okay, we can identify individual trees. Like I said, we can measure them. So what does that look like once we measure them? So here on the left is that image. Individual trees are just given a random color. We can measure trees in terms of their height and crown diameter of the crown volume. Now, taking those measurements are not particularly, and that relationship is not particularly groundbreaking. Larger trees typically have, or taller trees typically have a larger crown diameter and a bigger crown volume. But the slope and the shape of that relationship is fairly universal across the plots, right? So that can be used as a restoration sort of tool to track restoration through time. As far as our composition goes, we're currently using ground-based methods we're coupling ground-based surveys with our aerial imagery, both multispectral and hyperspectral imagery. So in this case, we have that same aerial imagery. We're segmenting out those trees, and we have our team of botanists on the ground who identify those individual species. That feeds back into an AI model so that this aerial imagery then becomes basically a very rich data set that you can, over time, should be able to identify those species as you go based upon the training data that's provided by ground-based surveys. So to put it all together, our goal is to attempt to cohesively measure across spatial and temporal scales to describe the structure, the composition, the function, resilience, and the sustainability of that reference ecosystem. Ground surveys are the gold standard, but project size and scope in this case demand moving to more appropriate spatial scales. And our overarching theme is to leverage emerging technology and analytics for faster processing and knowledge sharing, feeding back to management and decision making, and using tools such as bioacoustics, camera trapping, omics, eDNA, iDNA, and obviously drones like I've talked about. So thanks, and I'll take any questions.
Thank you, Dave. We do have time for a couple of questions. Does anybody have a question for Dave?
Thanks, very interesting. Question about your reference plots. Correct me if I'm wrong, but I think they were 1 hectare plots. Obviously, a lot of work went into those. My question is, how many reference plots were you interested in using as your baseline measurement?
I think we're currently at 12 or 14. the rarefaction curves in terms of species accumulation is just starting to show that we don't need too many more as far as that goes, but obviously structural, sort of components and sort of defining the whole range of whole range of structural and compositional sort of characteristics. You could probably keep adding and adding and adding, but there's probably a bit of a cost benefit that needs to happen there. How much time it takes to put in one plot versus what you get out of it.